Yeah, so we're recording this so that we'll have notes that way, but also people can put comments in chat and you can take screenshots, and we have this link group that you can post information in, or you can email comments or thoughts to the email address there, Monteleone Lab, and we'll consolidate them. Put CASP SIG in the subject line. So these are ways we can have some communication, and maybe we'll do something more formal in the future. So this is, the concept and the scope is that proteins and nucleic acids, biomolecules, molecules in general, live on a conformational energy landscape. They sample multiple conformations that we call the native structure. And they may even sample alternative conformations of higher energy states, which are very important to their function. In some cases, the conformational landscape has a lot of deep minima. In other cases, it might be quite flat, and the protein or biomolecule can adopt many, many conformations. We call this intrinsically disordered regions or intrinsically disordered protein structures. And this just makes the point that often the function of the protein depends critically on these alternative conformational states. And it also makes the point that when predicting alternative conformational states, we might be able to predict the two states, but not the relative energies between them. That is the Boltzmann weighted populations. And of course, ideally, you'd like to know the whole energy landscape and the differences in energies, but sometimes it's useful just to be able to predict that there are multiple conformational states that are among low energy minima. And so that will be part of our discussion, is the importance of getting Boltzmann populations relative to just being able to say there is an alternative state the biomolecule might adopt. This is just an illustration from some of our own work. This is the dengue virus protease, covalently bound to a lead compound for drug development. It was a project with Janssen Pharmaceutical. And the structure in solution using NMR methods, we could see there were actually two different conformations in dynamic equilibrium with one another. Here we just made just a morph between those two structures to see the differences. But it just illustrates the idea that proteins are not static objects. The target structure may be more than one structure, and that this can also be very important in practical things like thinking about a drug lead optimization. So we put together a kind of vision statement, and I don't mind you taking a screenshot of this. What is the point of this SIG? So the current vision statement, recognizing that biomolecules do not have static structures, but rather adopt a distribution of conformational states, the CASP Special Interest Group aims to evaluate if critical assessment of predictions of conformational ensembles or multiple conformational states of molecules is feasible. Is this something we can do in the context of a CASP experiment? So the SIG will consider developments in the fields of structural and computational biology that are anticipated in the next 2 to 5 years, and attempt to define protocols for assessing the accuracy of multiple or ensemble model predictions. And I think part of these discussions will be also computational methods, machine learning, or other methods, maybe molecular dynamics methods, for predicting multiple conformational states of biomolecules and methods of assessment. I probably should have put this slide earlier. This is the steering committee for this group that was put together by CASP people, by the CASP organizers. So Damiano Piovesan and Silvio Tossato at University of Padua, Emily Tai at NIH, Wim Rankin and Shoshana Wodak in Brussels. And we've been having discussions together with the CASP organizers how this might proceed. We want to expand this. We definitely need to add someone from the cryo-EM community. If you have suggested names, send them to this email address listed here. This is just to make the point that this question of modeling conformational ensembles, the scope is a continuous range of conformational distributions. The simplest thinking is that a protein may adopt two primary conformations, like these two structures, two crystal structures of adenylate kinase. And the challenge is to be able to model the fact that the sequence has these two conformations that can interconvert, and maybe even something about their relative populations. Or it might be three conformations or 10, and now becomes issues of, if you have enough data or to validate if you have the difference between 10 conformations or 8 conformations. And in the extreme, we're talking about hundreds of conformations potentially that might constitute unfolded or intrinsically disordered proteins. And one thing we need to discuss is, does this SIG really cover this whole range of ensembles, or is it more focused maybe at this end of the spectrum? And that's something for discussion. So an important part of this is it's not purely theory, it's not purely computation and machine learning. It's also thinking about how we can validate against experimental data. And what does that mean? Well, in X-ray crystallography, you sometimes will see alternative conformations in different crystal forms. And there are many examples in the protein data bank of proteins that have been crystallized in different conformations. Even within the asymmetric unit, you might see more than one conformation. Or sometimes you can fit the electron density to multiple conformations, which is kind of a little different aspect of how you can get it out of X-ray, get this information from crystallography. But we also have the question, do crystal lattice interactions shift the conformational distributions? Maybe they stabilize otherwise low populated states. Maybe they even induce structures that are not relevant, possibly. So cryo-EM is particularly recently able to generate multiple models of biomolecule structures from cryo-EM data. And this looks like a very promising area for getting experimental data to assess against predictions. And finally, NMR. NMR is in solution. There are several different kinds of parameters in NMR that are sensitive to conformational averaging and can be used to extract multiple conformational states, like I showed for that dengue virus protease. But there are challenges in terms of size and quality of data. And there are other experimental methods which also are sensitive to multiple conformational states, particularly for fluorescence energy transfer experiments or chemical cross-linking experiments, and also SACs. So the question is, can you actually do a CASP experiment with these kinds of data? So these are questions for our discussion, which will come after the presentation. You can take a screenshot of this as you like. I'll come back to this slide. Or we're going to discuss.
Instead of taking screenshots, is there any issue with sharing the slides afterwards? I think that would be more effective.
Yeah, absolutely. We can share the slides afterwards. We have to figure out how to collect a mechanism for doing that, but we will definitely share the slides.
Thanks. That would be awesome.
Yep. I'm taking a screenshot so that someone can look at it while even the presentation is going on to think about these questions. So we'll come back to these. What is the role of this SIG? What can it do? And what's the balance between modeling two states versus modeling intrinsically disordered proteins? Is it possible to organize ground truth data on alternative conformations? And how could that be done? How should this activity relate to other SIGs, like the SIG on machine learning and other aspects of CASP prediction or IDPs? And how will we go forward? So we'll come back to this slide. Now I'd like John, if you will, John, to take control. I think you can. And John is going to give us an overview of what was learned in the last CASP, or what challenges were encountered in this alternative conformation activity. Can you share, John?
Can you unshare first? Great. Okay.
Yeah.
It's working?
Yes, it's good. Very good.
Yeah.
Okay, so thanks, Guy, and the committee for allowing me to do this. First, let me say, I think it's great to see the SIGs, several of them now taking off. What we, the organizers of CASP, hope is that this is going to bring a more collaborative and new energy and leadership into what we normally do. So I see it as complementary to the main experiment. So everybody knows that there was a great upset in 2020 when AlphaFold 2 came along, and that upset extended to how we do CASP. It was clear that there were new areas where deep learning might start to have an impact, and we added 3 of those to what we're already doing. One was RNA structure, another was protein ligand interactions, and the third was ensembles. Now, this is very much a pilot, let me emphasize, and so I'm going to go through what happened, how we now think about it, to try and set the stage for the discussion of where to go next. So Guy just gave you his view of how to think about these kind of structures. I'm going to give you a slightly different one, which is highly CASP biased. I'm not saying this is some sort of absolute. I think it's an attempt to classify the different sorts of targets we might have in CASP. And so here it is, ensemble types. And as you can see, there are 3 main branches. There are ensembles which are present under fixed environmental conditions. So this includes—
John, can you make it bigger?
No.
It's
I'm going to blow up the bits that matter in a minute.
okay, thanks.
So this includes intrinsically disordered and other things that I'll briefly go over. The effect of environmental change on conformation. So I think that this turns out to be certainly within the scope of how we regard things in CASP. And finally, ensembles of uncertainty in experimental conditions. So this is really CASP specific, and I think to many of you won't be so interesting, but now in evaluating CASP results, we need to worry, because of the accuracy of many of the predictions, about obvious things like the effect of crystal environment on the selected conformations, about EM particle selection, what the resolution of the structure is, and so on. I'm not going to talk more about these, but if people have got opinions about it, be very happy to hear them. By the way, please use the chat. Please follow up with comments. The whole idea here is to provoke some kind of responses.
Yeah, and I'll just add there, we'll collect all the comments from chat together with the slides and the video and make them available. So if you put your comment in chat, it will become part of the record.
Okay, so I'm going to go through samples from these 2 branches where we've had targets and talk about them a little. So let me blow up this first fixed environment case. And so the blue dots are here where we either had targets or considered targets. So intrinsically disordered, by Boltzmann excursions, I mean regions of the conformational space which are explored around at least some nominal equilibrium conformation, either short range, sort of atomic vibrations that might be very important in, let's say, enzyme catalysis, larger range kind of normal modes explorations, and something which you'll see comes up, flat regions of the energy surface where there's KT or a couple of KT difference as you move conformationally. Also going to look at the type of thing Guy was mentioning just now, low population conformations, some of which I think might be accidental. There might just be a low population conformation present, which has not been selected against and just happens to be there. But there certainly are what I'm calling ghost conformations present in a particular reference state, which reflect other aspects of function. And obviously one wants to know about those. These are all kind of equilibrium situations. Something else that may come up, though, during folding is some kind of kinetic intermediate. And it turns out we do have one example of that in CASP, and I'll go over it. Finally, you can get a mixture of conformational states depending on how many other partners happen to be bound to some reference molecule. I'm not going to go into this one, except to say that we have one target in CASP, which is an RNA with proteins bound, and it's the same protein, and two sets of particles were picked. One set of particles has 4 copies of the protein bound, the other has 6 copies of the protein bound, and the RNA conformation is very different in the two. Now, nobody could do that in CASP. I think that it was the only and first RNA protein complex we've ever had, and it was beyond what most people can attempt. I think next time, if we had things like that, they would be interesting to go after. Okay, let me talk briefly about some of these. And I'm going to start with one we actually decided not to do, because that's relevant, I think, to the sort of considerations in this discussion. So this was a target from Paul Barbias, and it's to predict the conformational ensemble of this 17 residue stretch at the beginning of Huntington protein. So Huntington, as many of you will know, is a large, it's about more than 3,000 residue protein, which is involved in Huntington's disease through aggregation of these polygluon stretches. Most of us have short polygluon stretches here, and sort of less than 40 is fine, but these tend to get amplified in replication. And if you have more than 40, you can start to have trouble with the disease. The conformation of these is influenced, though, by the flanking bits on each side. And what Powell was looking at, this is actually from an earlier paper, 2020, in which he was looking at this particular construct, 80 residues or so, was the degree of helicity in these first 17 residues, which affects what happens with the polygluon. So the experiment here, he specifically labels each of the residues and measures chemical shifts. This is a chemical shift distribution. So this is the first 17 residues we're talking about. And you can see that the shift increases, which I understand corresponds to increased helicity as you go down the peptide. The data he offered us were for longer stretches of polyQ to predict what happens to this distribution in terms of predicted helicity. So what one would have to do is generate a set of conformations and compare them with this sort of signal. We decided not to do this, and this is a recurring theme, because often with these multiple conformation situations, you have rather low resolution data, and it's hard to know exactly what you should compare with. So I go into this just to provoke discussion about whether these kind of signals should be used. Let me go on to the second example from the fixed environment, and that is for the flat energy surface. This is a RNA structure from Riju and Rachel. And if I've understood this correctly, they have done 4 sets of particle picking. There are 4 electron density maps superimposed here. And you can see mostly the structures are similar, but this arm over here droops to different degrees in the 4 experimentally determined structures. We asked people to predict variable regions. We didn't tell them which it was. Nobody did very well. And the question here is, what did you expect? This looks like it's actually a continuum where 4 particle sets happen to have been picked. And if you happen to predict somewhere between 2, should that be considered a reasonable prediction? There's a big question, I think, about how you relate these kind of data to the sort of things that people may calculate. The third example, find my notes here, is for different states of a kinase. This is from Babas Kilodimis. This actually, what I'm showing you is again a published paper from 2020. What he has given us is newer data than this. I'm not allowed yet, it's still under wraps, to show you those newer data, so I'm going to explain it from this previous experiment, which is similar. And so the way this is done is to take one reference state of a kinase, this is actually Abel, and to measure chemical shifts for methyl groups, mostly buried methyl groups. And the way the experiment works, you can not only detect the main chemical shift, but if you're lucky, detect chemical shifts for different conformations of that methyl group. Now, how do you make sense of those in terms of alternative conformations? What Babas does is to compare with different conformations of the kinase which have been determined. In this case, 12 structures of different inhibitors. So you've got a set of secondary chemical shifts from methyl groups. As far as I understand it, you can calculate these from the other structures. And now you say which of the other structures best matches those methyl groups. And in this case, this was one of the other matching structures. And overall, he deduced that there were 3 populated conformational states, one which is the dominant one, which I think is this one, and then 2 others. And you can see these are very low population. The interesting thing functionally is that there are resistance mutants. Some of these are shown here. So this is the inhibitors actually are used as cancer drugs and don't work in the presence of some of these mutants remote from the binding site. And what the second part shows here is in the presence of this H415P mutant, how the third minimum disappears and you get more of a population of the second one. So it's very nice, right? It's a very nice idea and just the sort of data that we would like to have. What Babas gave us was actually data for 3 other kinases he's been working on, where he's done a similar thing, but actually instead of using a series of structures only from that kinase, he's comparing with other kinases as well. And so you say, well, I might have a representative conformation for one of these alternatives, which is actually not in the same sequence exactly. So we have all these coordinates. Dan, who was assessing this, looked at it. My understanding is the results are not very good. Nobody was able to get these alternatives. I think, and again, this is a subject for debate, one thing it would be good to discuss is, does it make sense in that situation to compare with coordinates? Shouldn't we be comparing directly with the methyl group chemical shifts? All right, so these are 2 sort of background things. Let's move on to targets where I can show you what's going on. And so this is a target from Ewan McRae. This is a designed RNA. The green is the mature structure. You can see it's sort of a bundle of helices, although there are a lot of, as usual with these RNA structures, specific details. And the brown is an early structure which persists for something like 8 to 10 hours after transcription. And so these are cryo-EM structures of both. You can see that the early structure has this helix in the end is sort of sitting in this groove a bit. And in the early structure, it's sort of bent over the top like this. I think people could predict the mature structure some, but nobody, as far as I know, got anything like this early structure. And I think what's interesting about this is that this may be many kilocalories higher than the final structure, and so out of range of something like AlphaFold 2 sampling. In fact, however, should say that for RNA this time, none deep learning methods are still the dominant thing. I think these are probably fairly unusual. It's a design, and so maybe it's not the sort of thing that ever happens in a natural sequence, but nevertheless, it's a very intriguing kind of challenge, which it's hard to see right now how people could be able to address. Okay, let me go to the second branch of the tree. And so now this again is when an environmental change produces a conformational change. And you can see we've got 4 samples here. I'm going to show you in detail the ligand binding, so we'll come back to that. Macromolecular binding, so now if you form a protein complex, of course, there are often major conformational changes, and I haven't actually pulled out an example of that because it works. I mean, maybe that's not very good for sort of pushing CASP results. I mean, the big triumph of this CASP, of course, was major progress in predicting macromolecular complexes, including when there are major-ish kinds of conformational changes. So we could put a tick on this one. Mutation effects, I'm going to come back to in a minute as well. We've got one very nice example of that. It's actually quite unusual that mutations produce big conformational changes, so we were lucky to get one. And then there are other things. I'm going to talk about this crystal environment effect in a minute, but we did not have anything in the way of post-transformational modification, for example, which would be really nice to have, something like phosphorylation that drives activation of a kinase. But of course, there are many things like that that might make good targets. Okay, let me show you several of these. Let me start with this crystal environment. I'm going to show you three, I guess. Let me start with this crystal environment. So this is an interesting protein. It's a coat protein from an insect virus, and it has a storage form in insect cells where it forms a crystal. These crystals are very stable, and they hang around for a long time. Actually, I understand the structure was done from crystals from the 1950s. They have very low solvent content. This is one of the only two structures in CASP15 where the current methods really produced bad results. For those of you familiar, the GTTS score of this, the best model is about 40, which is not random, but it's pretty bad. If you look at a comparison of the best model with the crystal structure, this is what you get. So here, the bluish color, turquoise, whatever it is the crystal structure, and the green is the best prediction. In fact, I think there are a lot of the predictions which are very similar. This is a sort of consensus structure for current prediction methods. You can see that this helix is only partly reproduced by the calculations and is substantially longer in the crystal structure. And it also has these strange bits coming off which don't interact with anything. Now, we don't have lattice information, but we're guessing that this actually is probably intimately involved in the lattice. And you can see, again, there are some regions which agree with the experimental structure, but not many. Green doesn't agree with turquoise experiment. And there's one region of the predicted structure, which is actually quite long, where the methods don't know what to do. So this is a sort of unpredicted region. And we would guess that in the crystal, this probably does something. So two ways of looking at this. You could say, oh, this is a crystal artifact, and maybe the calculated structure is actually better in representing in vivo conditions than the experimental structure we happen to have. But of course, as I mentioned, this is also a structure which is in a coat protein, a polyhedral coat protein for a virus, and probably doesn't have the solution structure there either. So there are three different structures: what goes on in solution, what goes on in the crystal, and what goes on in the viral coat. And sorting out those kind of situations is obviously still a very challenging thing to do. All right, I've talked about things that didn't work. Let me finish with two examples of things that really are quite pleasing in that they did work somewhat. The first of these is a type 4 ABC transporter, where by the way, these are the acknowledgements to the people who submitted these targets, where we were given actually 5 sets of coordinates. One is the apo structure, that's the green here, and the other 4 are for different ligands bound. I think you can see this for this ligand. Obviously, there is a closing. This is a single chain. There's a closing of the interdomain angle here in response to the presence of this particular ligand. Altogether, there seem to be 3 different conformations, the apo one and 2 different closed ones. There's an even tighter sort of angle in a different ligand. And although we have not, to my knowledge, got a detailed assessment of this yet, it's clear that some people were able to predict these angles, at least approximately. And I think the reason for that, looking at what people did with ligands more generally in this CASP, is to use templates from the PDB. I don't know this. We still have to verify this with the people who do dwell on these. But judging by what was done for other targets, there are probably examples in the PDB with similar ligands and similar transporters from which you can deduce what the likely angle is. Again, for ligands, deep learning methods were not yet the most effective. So it's these more traditional and actually quite powerful methods which are giving nice results. So for this very amazing looking system, some things can be done. Finally, let me talk about the single mutation case. It's a little bit complicated, so I'm going to do it in 4 slides. This is the experimental structure. It's an enzyme, an isocyanide hydratase. It's a dimer, normally a symmetrical dimer. So here's one of the monomers, here's the other. There's an interface, of course, here. And what I want to draw your attention to, if we look at the top monomer, these N terminuses here go around. This is coming towards the C terminus. And then this bit, which I've colored brown, is the last piece of the chain. And you can see it comes across, interacts with the other monomer, and then packs against its own monomer at the last piece of the chain. The mutant that was introduced is here in the wild type, and this is what I'm showing you, is an ASP, and it was mutated to an alanine, which disrupts, apparently, actually I have to say this doesn't make entirely energetic sense to me, but I assume it's right, disrupts interactions with this piece of the chain. But what happens is interesting. Just before I go to that, so this is symmetrical. Here's one of the C-terminal tails, and of course here is the other C-terminal tail coming over to interact with itself at the end and with the other monomer in the middle. Now introduce the mutant, and that brown tail, instead of going over here, I've now colored it red, turns and goes where the green tail of this one was in the previous structure. So it's a sort of domain swap. It makes the same interactions which were self-interactions here, but now they're inter-subunit interactions. And of course, the other tail does something similar, except interestingly, symmetry is a little bit broken. You can see that this is not quite the same as this. Now, amazingly, several groups did a good job on this. Let me show you. I'm going to switch to a monomer representation because it gets very messy when you superimpose all these things. Sorry, you can skip that. So here is in green, the experimental structure, in purple, the prediction. I've taken a slightly different angle, but this is the reference helix going into the terminus. The green then comes around. We saw this just now. This would be interacting with the other monomer. And you can see the purple, although it's not quite right here, does come around and does make the same interactions with the other monomer. Now, looking at, again, we have not verified this, but looking at the groups who were successful with this, they're all groups who use a lot of sampling, but basically are using AlphaFold 2. So it appears that AlphaFold 2 was actually able to handle this situation given sufficient sampling of the mutant, which is really pretty amazing, I think. Okay, that's basically what we saw in this CASP. Just to recap a little bit, a major issue that comes up is the low resolution nature of the data one often has and how one should best handle that, and whether or not one should be using coordinates to check these kind of things, or choosing systems where one can more directly compare with the experimental data. All right, I'm going to stop at that and leave it to Guy to open the discussion.
Thanks so much, John. That was great. And it's a great way to open this set of meetings. Let's see if I could share my screen again. And it actually makes me realize that, you know, the issue about comparing against coordinates versus comparing against data, something we discussed, but I didn't include in this list of questions. And that certainly is something we'll want to try to address. So I think the best way, there's a whole bunch of comments that have been put into the chat, which we'll capture. And in the course of this discussion, we'll talk about a little bit toward the end of the hour, how to distribute what we collect in these discussions. But I think the best way is for people just to make comments on John's talk or on the questions that are here, which we'll capture by recording the video. I should be able to see when people raise their hands. Can someone raise their hand? I should be able to see if I could see that. Otherwise, I think we might have to have people just commenting and see how it goes. So I see 2 participants raised hands. Okay, so let's start with Arnie Ellefson. Go ahead, Arnie.
No, I was just promised. Shoshana says she was kicked out because there were too many people here. So at the moment it seems to be limited to 100 people.
Oh,
and
look at that.
That she was kicked out. I don't know if you can do anything about it, but that's the case.
Thank you. Okay.
99,
yeah.
there's one slot available.
Yeah, I mean, she's tried to rejoin, she said. I don't know, yeah.
I didn't realize we'd have such a good turnout, and I actually didn't realize there was a 100-person limitation. I'm sure I can change that next time.
Yeah, well, I guess it's next time. Yeah. No, I mean, I think we should do as much comparison with data, real data, experimental data, than with structures, because really, in many cases, structure doesn't make sense, because if it really depends on how the experimentalist or whoever made use of experimental data actually produce a structure. So certainly, if you have an NMR structure with 2 distinct conformation, it really depends on how you weight them and how you select them. When you cryo-EM, how many different ensembles do you pick up? It's clearly, I mean, it's a decision made by someone, and it's impossible for us predictors to predict that decision. So I think wherever is possible, we probably should generate structures that then are used for see how well they fit the experimental data and not how well they fit the structures. That I think is the way we should do it. Mm-hmm. I know it's not always trivial, but it's I mean, you can predict chemical shifts, you can predict NOEs and things like that. It's at least it's a try.
And it also is a nice chance for the experimentalists to learn from the theorists, and the theorists to learn from the experimentalists about the experimental data and how it relates to predictive models. Because you have to learn how to go to data from a model in the course of it. Yeah, so maybe next up, I'm just looking on the list here. How about Jens Myler? Hello, Jens. Do you have some comments?
Hey, good to see everybody. Yeah, just following up with what Arne said, I also think it's going to be key to move to other experimental technologies to sort of define the baseline here. And it's going to be very important to be inclusive. There are a lot of technologies that give structural data. There will be some challenges. One of the challenges is to it's better to compare your ensemble models to raw data than to data that in any way has been translated to necessarily structural information. We know that for NMR and EPR and other technologies. There will also be the challenge that most of that data are going to be limited. It will not be at atomic detail accuracy. So how do you judge your models if you don't have that level of comparison? And then unfortunately, what interests us from a biological point of view is not necessarily what these data are always good at measuring. So I think we need to sort of find out what are the biologically significant questions we can ask where we actually can collect the experimental data to compare our ensemble states or conformational switches, can compare those two, and that will actually be quite a challenge to might be on a case-by-case basis.
Yeah. So I'm going to talk less and have more people have a chance to talk. So Vladis Oliankovis, do you want to have a comment?
Yes, it's a bit similar to the previous two, but also just want to echo the importance of trying to match the ground truth closer to the actual observable. So while the three-dimensional model is nice and clear, but when we talk about ensembles, it's really usually some restraints or pairwise matrix that we can probably propose as a ground truth rather than coordinate. So kind of like I think we then should rephrase also some of the challenges to kind of given the sequence predict the pairwise matrix, or given the sequence predict provide the restraint between this and that. So and then also then talking about certain X-ray limitations. So we do know that crystal packing can affect the result significantly. So I think when in the future, when we provide some of challenges, it would be important to include the experimental data as part of the prediction. For example, if we say we have a crystal structure, then maybe we can also provide information what is the unit lattice composition and what is the symmetry of a unit lattice as well, and what kind of like under what condition has been collected, in such a way also to inform potentially if we try to predict closer to the experimental data.
Well, thank you. I see Wim Rankin has a comment.
Yeah, thanks, Guy. I just wanted to make the point that we also have to be careful to distinguish between a change in conformation by, for example, a mutation or a change in environment or PTM, for example. And so that part, I think you can in principle cover with coordinates as long as there's distinct conformations in those two different states. And the multiple conformational states that you might have in one of these states, right, which is not quite the same thing, because I mean, I think the second lends itself much better to direct raw data comparison, whereas, you know, the switches in conformation because of some change environmentally, post-translationally, you know, amino acid wise, it's more traditional, I guess, in the way that you can capture that in CASP. But I mean, it's a different thing to me personally, I think, and we should maybe distinguish between those two as well. And the list that you gave, Sean.
Yeah, I think that's a great point. The problem, though, is that you know, look at people who study conformational ensembles with WET or with IBBR, right? They introduce a bunch of mutations, add a number of tags to report the conformational changes, and then all the data on all of these different species is dumped into one model. So I think your point is great and very important, but one of the limitations of this is that a lot of the experimental technologies to monitor conformational ensembles actually intrinsically assume, you know, do the experiments on a whole bunch of, in principle, different species, and then assume they all report on sort of the wild type structure, which is amazing, but that's the, you know, state of the art still.
Yeah, no, it's messy. I agree. I mean, it's you know, but the conceptually, it's a different thing, I think, because you change the energy landscape. Like, are there multiple conformations within the same free energy landscape in the same condition? That's different from changing the energy landscape and observing a different conformation, essentially, right? I mean, that's the underlying kind of principle, I think. I just also wanted to quickly make the point, something else that might be interesting to look into, John, also from your perspective, is what you get with an ancestral protein reconstruction and the conformations there, because sometimes you can see that there's different lines that have specifically very different conformations, or like slightly different conformations, specific points, changing the function of the protein. Those are also nice cases, I think, to look into, because there you can, you know, have a protein with a slightly different sequence that is very similar to something else that's already known, but it might have actually a distinct conformation in a certain area, which makes it extra difficult, because AlphaFold will probably just ignore that because it will use pairwise information. So, I mean, that might be also interesting.
I think that's quite an interesting point. Go ahead, John.
Probably no, we did have a target like that in this CASP, an ancient sequence, so a restricted number of amino acids, 48 residues long, and there were two versions which formed different dimers. There were 5 mutations between the two. I didn't talk about it because at some point you're really talking about different sequences, right? It's, you know, whatever it is, 80% identical or something. I didn't know where to draw the line there, but you would say that should be included in this.
I don't know. I mean, they are fold-wise related, right? It's like
Yeah.
small changes in the conformation that can then have a functional effect, which is also interesting from that perspective, I guess, maybe. I don't know. I just thought I'd mention it.
Yeah, I guess one way of framing this, and I think somebody already emphasized this, is which of the conformational changes are actually functionally relevant? And so, whether or not you're talking about an ensemble under fixed conditions or changing the conditions, the key thing, or one of the key things, is has that functional consequences? It seems to me. But I'm listening.
Well, a point I want to bring up here, not to change move too far away from that point, is to what degree should this SIG span this whole range of the problem of validation of prediction from 2 states to intrinsically disordered states? There are a number of other SIGs on intrinsically disordered proteins. But what's unique about this is the connection with CASP and the prediction community. So should this be part of our thinking, or is it really outside the scope of what we can imagine to do with the CASP experiment? Oh, I see some hands raised. Go ahead. Was that Arnie?
I think that CASP most likely need to limit ourselves to the actual have some structural data. So if you have an intrinsically disordered protein that somehow can be in some environment get a structure or a part of the structure, then it can be included. But if it's just disordered, we don't have any structural information, we just have some sequence motifs, limbs that are binders or something, that's certainly a quite different beast. So that's certainly interesting, but then you're predicting binders basically out of that. So I think we need to sort of stick to the ones where we have structural data. And most intrinsically disordered proteins we don't really have any structural data for. So that's would exclude quite a lot of them, I think.
I'm just going to look at the raised hands. I see Vladis Oliakobis also has a comment here.
Yeah, just also that I think there's also a typo inside intrinsically disordered proteins. I think was intended.
Yeah, you're right. It's a typo in the slide. I'm sorry. I'll fix that. Thank you.
No worries. But it's also I agree. But if a protein has only a partial part that is disordered, I think it shouldn't disqualify it. Especially like linkers, they often tend to be a bit kind of like loose on the data. But I think we can probably limit ourselves to some definition of structure prediction, and that at least some parts of it, or it has some clear populations that are ordered, and I think that would be a bit more kind of like also restrictive. I think cryo-EM structure is also like an interesting approach, interesting piece, because with NMR, one of the limitations that we probably can study only a limited size of proteins, whereas with cryo-EM we can get a lot of these particles and density envelopes that we can reproduce. So again, that could be an like if you look at it as can you, given a sequence, can you predict what would be the likely average envelope of this? and kind of like in a way, again, trying to bridge it back to the observable and basically predict the experimental observable of that for a given condition.
So what is the resolution of an envelope for a disordered protein? I mean, do you really, I don't know this.
Oh, for the disordered proteins, like I think you would struggle to get an envelope, so that would also
that's my thought also.
help you to define what are the things that we likely would be able to predict and would be likely to be compared with observables versus something that is kind of like basically anything works. Because I think with disordered proteins, if the best thing we can do is some radius of gyration, then it's really not a very precise way of measuring success or kind of even going back to the function of the protein itself, because there could be so many wrong solutions that could fit that variable. So I think we want to have something a bit more precise on the given residue level, pairwise residues or something. So yeah, So if part of a protein is disordered, but majority of it is ordered, I think it's fine. But if it's kind of like completely disordered, I think it's very little structure to be predicted.
Shoshona, do you have a comment? See your
I
hand
just
up.
came back from being ousted from the meeting. So I think, one case that could be actually considered is if a disordered protein actually binds to an ordered domain. And these type of targets, we had in Capri. And, you basically don't tell which part is binding. And you have to predict, the bound structure of the disordered protein. I mean, that's a case where this is doable because you basically, have actually the structure of the bound piece.
Mm-hmm. Jens Meiler?
Yeah, maybe I just add to this discussion. I think we might want to separate us a little bit from the term intrinsically disordered protein, because I think it's established, but it's to some extent ill-defined, right? We usually talk about these proteins when they adopt some function, and that function usually involves creating some sort of structure interaction, secondary or tertiary structure at that moment. Yeah. And this is when we collect the experimental data. So again, I think it would be better to think about functional relevance. And if there is a protein that people call intrinsically disordered, that assume a concrete conformation or ensemble of conformations at the moment they adopt function, that is the moment when we have a chance to collect experimental data. And this is a moment when we can make a prediction and compare it against those data. I think nobody here is thinking of a peptide that is in its entire life entirely random. I mean, that obviously is probably biologically not what we would want to be interested in the first place.
So I think this was a great discussion, and I very much appreciate John leading off this SIG and giving us the right vision of where we can take it. And I thought the discussion was very valuable, and a lot of good ideas were put out. What we're going to do is between the video and the notes and listening to what's in the chat, I'll work with the steering committee to put together a set of notes from the meeting, and that will be available together with the slides. I have to figure out a way to put together a mailing list, and we haven't created a registration site yet. So I think if you send me your email to our lab address, I can start putting together an email list to share information as we collect it. But then soon we'll put out a registration site, and maybe in a few weeks, and so we could collect emails that way. The plan is to do this every month, but we decided not to have the meeting in April. They're going to be the first Wednesdays of each month, and in that overlaps with the Passover holiday in April. So we decided we can just digest what we got and have our next meeting the first Wednesday in May. And Professor Shoshona Wodak has agreed to talk about a database of alternative conformational states, or looking in the PDB and organizing alternative conformational states as a benchmark. And so that will be the presentation that day. We want to also cover methods. We have in this group here some of the leaders in developing methods for modeling alternative states with modifications and variations of AlphaFold and other methods. And so we've been looking at some of those papers, and we may invite some of those people to do a presentation. But if you have suggestions for presentations or things you'd like to present, send them to this email address, and then we'll organize those. And finally, as I said, if there's somebody else who would like to join the organizing committee or want to propose somebody, you can also send those nominations. So we're pretty much up to time. We have a few more minutes. I want to stop right on time, but does anybody else have other comments before we close off here?
One practical comment, Guy. No, raise your limit of Zoom. above 100
Yeah.
because I couldn't get in again. for
Yeah,
most
I haven't had
of-
that problem before, but that's something we'll do for the next meeting. I'm sorry about that, Shoshana.
It's okay.
I'm sure
no,
there's
no
a lot of
problem.
other people who got
Hey,
turned
Yeah,
away
yeah,
because of that.
hey, Guy and John, thanks so much for putting this together. This is awesome.
Yeah, thanks.
I have a couple of comments. I think one comment is I do think to really include this in CASP in an efficient way, we need to think about what kind of experimental data we want to compare to. And that is really there's a wild zoo of opportunities here. And I was part of I mean, you guys know this, right? Andre Charlie led this task force. There's the PDB dev where people make sort of a concerted effort to look at some of these other technologies to collect these data and created a portion of the PDB that collects some of these models. So it might be entirely worthwhile to also maybe include some of the leaders in the field that collect these type of data that might be at the point where we can really use them for comparing models to, and maybe also coordinate with PDB dev in particular if we're talking about creating a database of alternative conformations. That's super exciting. That's a little bit different what PDB dev stands for, but maybe talking about synergies in that area would be worthwhile.
It's a very important point, Jens, and maybe we'll in fact have a session beyond Shoshona's on the kind of data that might be put together and what's feasible to do for a CASP experiment. It's one thing to take data that's out there and benchmark with it. It's another to have blind data that people can use in CASP. That's much harder. And that's clearly a goal, a challenging goal for the SIG, is to come up with a plan for that. So I'd say probably later in the spring, we'll have just the kind of discussion you suggested.
Yeah, if I could just follow up on that, I think from a CASP point of view, even though it's a year away from the next prediction season, as it were, this clearly isn't going to be trivial, and sorting out which sorts of data we should be looking for and started beating the bushes for possibilities is actually fairly urgent. Like 2 months is fine, sort of thing. But the other thing is, I think it would be a pity if the discussion on this group was restricted to what, I don't think Guy intends that, but is restricted to what fits in traditional CASP. I mean, maybe there are other things to do here, like Shoshana's benchmark, which are a better approach or a complementary approach to what CASP tries to do here.
Yeah, I think this is an open discussion. What can be done, now, and what kind of benchmarking can go on.
Okay, we're going to keep to time because we're able to this time, and please email me if you want to be added to the email list based on that address I put out, the lab address, and we will be in touch with people. So thank you very much, everyone.
I'm sorry, can I have one last
yes,
question?
go right ahead.
Sorry for interrupting, but I mean, I really think this will be one and maybe the most important next challenge in protein structure prediction. And I really wonder, I mean, this looks like you have an incredible turnout. Is there a plan to have a more like in-person, longer kind of conference style meeting for this community to meet and form? Because I think this is gonna keep us busy for a few years.
I think that's certainly a vision and a direction. Right now it's a special interest group to begin to brainstorm. It looks like there is a good interest, and maybe there'd be a chance to find the funding or a mechanism to organize an in-person meeting. But I think that would be there have been a few. It's been the topic of sessions at some different meetings, but I think that you're right, Jens. I think this is the exciting next challenge in the field of structural biology.
Maybe I can add that, this has been a major activity, in Elixir here in Europe. We have been looking at this and trying to, this is something that really is on my mind and the mind of many of Capri, Capri predictors. It's a huge problem that kind of haunts us, for many, many years. And now is a possibility maybe of having some breakthrough, especially also in simulation methods, etc. So it has a very wide, wide reach in terms of methodology.
Well, it's exciting to see how much enthusiasm there is, and it's great that we had a good turnout. So we're going to end here, and we will meet again on May 3rd. Okay, thank you. Bye.
Thanks,
Thank
guys.
you. Thank you, everyone.